the 2013 paper. So starting with the anatomy, first carpal bone to appear is, so first carpal bone. What are the carpal bones, students? Carpal bones are the bones which are present in your wrist. Okay, which are present in your wrist. The first carpal bone to appear is the capitate. So answer is B, capitate. The ossification center in carpal bones appear as first capitate, then triquetrum, then scaphoid, then pisiform, then hamate, then lunate, then trapezium. Okay, first will come the captain in the first year, 1 year. Okay, first capitate will come, captain will come, then triquetrum, then scaphoid, then pisiform. Then pisiform, okay. Then pisiform will appear. Fine. First captain will come, then you are with captain Trichela, that is triquetrum, tri, then scaphoid and trapezoid, pisiform. Pisiform is at the last. See, first capitate will come, sorry, there's some printing mistake here. Capitate will come first, then hamate will come. Capitate with helmet will come, then triquetrum, then lunate, then scaphoid and trapezoid, then trapezium, then at the last pisiform, because pisiform is the smallest one, right? Pisiform. It is the ossification of the tendon, pisiform bone. Okay, let's see next. First captain with helmet, hamate, then trinchula, triquetrum, then lunate, then scaphoid, trapezoid, trapezium, then pisiform. Okay, if you are enjoying the session, give a thumbs up. Pharyngeal muscles are derived from which pharyngeal arch? Important question, okay? Pharyngeal muscles. Muscles which are present in your pharynx, these are derived from which pharyngeal arch? These are from second pharyngeal arch. Pharyngeal arch, pharyngeal muscle. Pharyngeal muscle is third arch. Spinal cord develops from. Spinal cord, it develops from the neural tube. So answer is neural tube. Nervous system develops from ectoderm, neuroectoderm. So nervous system develops from neural tube, which in turn develops by process of neurulation, that is formation of neural plate and its enfoldings into neural tube. Structures formed from neural tube are from cranial part, enlarged cephalic part give rise to brain. Developmental parts are forebrain. Forebrain is also called prosencephalon, mesencephalon, midbrain. Hindbrain is called rhombencephalon. Forebrain has parts like telencephalon and diencephalon. Telencephalon is cerebral hemispheres and lateral ventricle. Diencephalon is optic cup and stalk, pituitary, thalamus, hypothalamus, epithalamus, pineal gland, and third ventricle. Midbrain, cerebral aqueduct. Hindbrain, rhombencephalon, cerebellum. Metencephalon, cerebellum pons. Myelencephalon, medulla oblongata. From caudal part give rise to spinal cord. Okay. Collecting part of kidney develops from. Collecting part of kidney. So kidney is developed from the ureteric bud. It develops from the. This is the embryology part. Embryology is important thing. Okay. So ureteric bud, it gives rise to the collecting part of kidney. So kidney is developed from two sources. Kidney is developed from two sources. If I am audible and fine, please give me a thumbs up. Like the video, share it, make a big circle. Collecting part of kidney is developed from. So collecting parts of kidney is developed from ureteric bud. So how do the kidneys develop? In short, I am brief. I am going to say kidneys develop from two sources. What are the two sources, students? Metanephrons. Metanephrons, metanephric mesoderm blastema is the lowest part of nephrogenic cord, which is derived from intermediate mesoderm. From the excretory unit of kidney, glomerulus, proximal convoluted tubule, loop of Henle, distal convoluted tubule. So metanephros, that is metanephric mesodermoblastema, the lowest part of nephrogenic cord which is derived from intermediate mesoderm from excretory unit of kidney. Uretic bud, it arises from lower part of mesonephric cord. So lower part of mesonephric cord, what you get? You get the uretic bud. It forms collecting part of kidneys, pelvis, major calyces, minor calyces, collecting tubes, and ureters. Okay, metanephrosis, metanephric mesodermoblastema, lowest part of nephrogenic cord, which is derived from intermediate mesoderm from excretory unit of kidneys. If you want to learn with the help of analogy, I'll ask Gemini to help. Okay.
The development of the kidneys in vertebrates is a fascinating journey that unfolds in a series of three overlapping stages, each characterized by a more advanced kidney structure. These stages, appearing in a cranial to caudal, head to tail sequence, are the pronephros, the mesonephros, and the metanephros. All three originate from the intermediate mesoderm, a layer of embryonic tissue. Here's a breakdown of these stages. 1. Pronephros, the pioneer, week 4 in humans. This is the most primitive and the first kidney to develop. In humans, it appears in the neck cervical region of the embryo around the beginning of the fourth week of gestation. It consists of a few tubules called nephrotomes that join with the pronephric duct. The pronephros in humans is non-functional and begins to regress by the end of the fourth week. However, the pronephric duct persists and extends towards the tail end of the embryo. 2. Mesonephros, the interim kidney, weeks 4 to 8 in humans. As the pronephros regresses, the mesonephros develops just behind it, in the thoracolumbar region, chest and lower back. The pronephric duct extends backward and induces the formation of mesonephric tubules from the intermediate mesoderm. Each mesonephric tubule develops a capillary network called a glomerulus at one end, which is enclosed by a Bowman's capsule, allowing for blood filtration. The filtrate then flows through the mesonephric tubule and drains into the original pronephric duct, which is now called the mesonephric duct or Golgi duct. The mesonephros functions as a temporary kidney in the early embryo. In males, the mesonephric duct plays a crucial role in the development of the male reproductive system. By the 8th week, most of the mesonephric tubules degenerate, but the mesonephric duct persists. 3. Metanephros, the definitive kidney, week 5 onwards in humans. The metanephros is the final and permanent kidney in reptiles, birds, and mammals, including humans. Its development begins around the 5th week of gestation in the pelvic region of the embryo. The metanephros arises from two key components. Ureteric bud. This is an outgrowth of the caudal end of the mesonephric duct. It elongates and penetrates the metanephric mesoderm. The ureteric bud undergoes branching to form the ureter, renal pelvis, major and minor calyces, and the collecting ducts of the kidney. Metanephric mesoderm, or blastema. This is a mass of mesenchymal cells that surrounds the ureteric bud. Through a process of reciprocal induction, where the ureteric bud and metanephric mesoderm signal to each other, the metanephric mesoderm differentiates into the nephron, the functional units of the kidney. This includes the glomerulus, Bowman's capsule, proximal and distal convoluted tubule, and the loop of Henle. The first glomeruli appear around the 9th week in humans, and nephrogenesis, the formation of new nephrons, is generally complete by 36 weeks of gestation. Initially located in the pelvic region, the kidneys ascend to their final position in the lumbar region of the abdomen between the 6th and 9th weeks of development. In summary, kidney development is a complex process involving the sequential formation and regression of three distinct kidney systems. The metanephros, arising from the interaction of the ureteric bud and the metanephric mesoderm, ultimately develops into the permanent, fully functional kidney.
Okay, so this is the complete detailed description of the development of kidneys, embryological development. So first, pronephros are formed, but in the 4 weeks of gestation. But these are formed in the neck region, cervical region of the embryo. Okay, it consists of tubules, few tubules. These tubules are called nephrotomes. The join with the pronephric duct. Pronephros in human is non-functional, begins regress at the end of 4th week. So then they will end, okay? persist and extend towards the tail end of embryo. Then by the 4 to 8 weeks, around 4 to 8 weeks, mesonephros is formed. As the pronephros regress, mesonephros develop just behind it in the thoracolumbar region. The pronephros were in the cervical region, then this mesonephros is the thoracolumbar region. Pronephric duct extends backward, induces the formation of mesonephric tubules from the intermediate mesoderm. From intermediate mesoderm, what happens? Pronephric duct extends backward and induces the formation of mesonephric tubules. So pronephric duct, which is there now, it extends backwards and induces the formation of mesonephric tubules. Then after mesonephric tubules, capillary network will form, glomerulus will form, Bowman's capsule will form, and allofiltration of the blood. Males, it will form the reproductive system. Okay. Then talking about last, metanephros kidneys. These are the definitive kidneys, and these are formed by the 5th week onwards. Okay. Metanephros is the final permanent kidneys. Okay. So these metanephros, they are two key components. One is ureteric bud. This ureteric bud, this is an outgrowth of caudal end of mesonephric duct. It elongates and penetrates the metanephric mesoderm. So this will form the ureter, renal pelvis, major, minor calyces, collecting ducts of kidneys. Then metanephric mesoderm. This metanephric mesoderm or blastema, these will form the Bowman's capsule, nephrons, glomerulus. Includes the glomerulus, Bowman's capsule, proximal distal convoluted tubules, and loop of Henle. Okay. The first glomeruli appear within 9th week in humans, and nephrogenesis, the formation of new nephrons, generally complete 36 weeks of gestation. So initially located in pelvic region, then kidneys ascend to the final position in the lumbar region of the abdomen in 6th and 9th weeks of development. The metanephros arising from interaction with the ureteric bud and metanephric mesoderm. Okay, so this was the complete description we got here. Next, which level the somites initially form? Somites initially form in the cervical layer. So answer is B, cervical layer. The first pair of somites develop a short distance posterior to the cranial end of the notochord, and the rest of the somites of the caudal. Okay, by the 20 days, the first pair of somites have formed in the neck region. So which level the somites initially form? At the neck region. So paraxial mesoderm differentiates into somites. So dermatomytome gives rise to skeletal muscles and dermis. Sclerotomes give rise to vertebral column. Paraxial mesoderm differentiates into somites. Okay, by the end of 22nd day, the first pair of somites have formed in the neck region. After there's about 3 pairs of somites are formed per day, and at the end of 5th week, about 40 to 44 somite pairs are formed. 4 occipital, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, 8 to 10 coccygeal. Differential dermatomytome give rise to skeletal muscle and dermis, as sclerotomes give rise to distal column. I don't know anything. Are paired blocks of mesoderm. So these are blocks of mesoderm. One of the 3 primary germ layers in the early vertebral embryo is the mesoderm. So they form along the anterior posterior axis, head to tail, on either side of the developing neural tube and notochord. So this is the notochord or neural tube. So their blocks are formed. These are called somites. These are called somites. Okay, complete description here. Formation somatogenesis. Somites arise from the paraxial mesoderm. a band of mesoderm located adjacent to neural tube. Okay, band of mesoderm which is located adjacent to the neural tubes. The formation of somites is a highly regulated and rhythmic process called somatogenesis. Begins in the anterior part of the brain, sequentially towards the posterior. New somites bud from the anterior end of the presomatic mesoderm, which is the unsegmented paraxial mesoderm at the tail end of the embryo. The timing of somite formation is species specific. Approximately forms every 90 minutes. So somatogenesis is controlled by complex interplay, segmentation, clock, and determine all these as somite cells within the undergo mesenchymal. This is not about structure and differentiation. Each newly formed somite is initially a relatively uniform ball of cells. However, as the somite matures, cells undergo degeneralization and differentiate distinct components. Sclerotome, dermomyotome, dermatome, and myotome, and syndrome. See what are these? I was looking for this for the expression here. However, the somite mature itself undergo regeneralization and differentiate into distinct compartments. Sclerotome. What is sclerotome? This is the ventromedial part of somite. Means ventromedial means close to the neural tube and notochord. Sclerotome cells undergo epithelial to mesenchymal transition, migrate medially to surround the neural tube and notochord. They will eventually differentiate to vertebrae and rib. So what they do? They migrate and surround the neural tube and notochord like the vertebra. Dermomyotome. So this is the dorsolateral part of the spine, remains epithelial for a longer period and further divides into dermatome and myotome. Dermis, connective tissue. So the outer layer of dermomyotome that contribute to the dermis is of the dorsal skin. So dermis is skin. So it gives rise to dorsal skin. Myotome means inner muscle forming portion of the dermomyotome further subdivides into epaxial myotome forming the deep muscles of the back and hypaxial myotome forming the muscles of the body wall, limbs and tongue. Okay, hypaxial body wall, epaxial deep muscles of the back. Syndrome, formation that appear transiently and contribute to the formation of tendons. Okay, development and establishing the segmentic pattern of the vertebral body. They are the precursors for a significant portion of axial vertebrae and ribs. Skeletal muscles in the dermis of the back somites also play a crucial role in guiding the migration of neural crystals and axons, and of ensuring the proper organization of the nervous system and the body axis. Form is relatively conservative species and can be used to determine the embryonic age. Can lead to congenital abnormalities, spinal musculature. Okay, so which level the somites first usually form in the cervical layer? So the dermomyotome, sclerotome. So the sclerotome form the vertebral column, skeletal column, and dermomyotome, myo means muscle, dermo means skin. So it will form skin and muscle. Double inferior vena cava is formed due to. This is all geology questions I have asked. A lot of geology questions. Okay. Double inferior vena cava is formed due to. So double inferior vena cava. Okay, I should zoom it. This is the maximum I can zoom. Yeah, this is the maximum. So double inferior vena cava is formed due to. So it is formed due to sarcocardinal veins, no. Supracardinal, subcardinal, no. Persistence of supracardinal and subcardinal veins. Developmental anomalies of veins. So first you will understand the development. Double inferior vena cava is formed due to. Okay, so these are anomalies. Vena cava is the largest vein in the human body and is responsible for deoxygenated blood from the body to the heart. Okay, it is actually composed of two major vessels, superior and inferior vena cava. Superior vena cava formation. So from the upper half of the body. Okay, so it's not explained in biology here. But let's understand this, then we move to embryology part. Okay, superior vena cava collects deoxygenated blood in the upper half of the body above the diaphragm, including head, neck, upper limb, chest. It is formed by the union of left and right brachiocephalic veins. Each brachiocephalic in turn is formed by merging of two smaller veins. Subclavian vein drains blood from upper lip. Internal jugular drains from the head and neck. So there is the internal jugular from the head and subclavian from the upper lip. So they together join and form brachiocephalic vein. Brachio means here limb, limb. Cephalic means here head and neck part. So the together brachiocephalic, brachio is subclavian vein and cephalic is head and neck vein. That is the internal jugular vein. So internal jugular vein and your subclavian vein. Yes, subclavian vein this form brachiocephalic vein. This will further form superior vena cava. So union of left and right superior. The left and right brachiocephalic vein, they will form the, because from the right arm and left arm, both arms, right, and from internal jugular vein, so they together will form the brachiocephalic. They together two brachios, left and right brachiocephalic vein, form the superior vena cava. Left and right brachiocephalic vein join posterior to the inferior border of first right costal cartilage. So first right costal cartilage forms superior vena cava. As we see, then descend vertically and enters the right atrium of the heart. Okay, talking about IVC, inferior vena cava. So lower half of the body, it collects blood, including legs, abdomen, pelvis, joining of right and left. But for superior vena cava, brachiocephalic veins, brachio hand and cephalic neck for joining. But for inferior vena cava, it's not collecting blood from head and neck and arms. It is collecting blood from legs. So it is iliac veins. So it is abdomen and pelvis. So it is iliac veins. So it is formed by right and left joining of iliac veins. Each iliac vein is formed by the union of internal iliac vein, which drains blood from pelvic organs and gluteal region and pelvic organs, whereas lower limb say external iliac from the lower limb, internal iliac from the pelvic and gluteal. Right and left common iliac veins merge anterior to fifth lumbar vertebra to form inferior vena cava. So fifth lumbar vertebra. Okay, fifth lumbar vertebra to form inferior vena cava. So the IVC that runs through the abdomen alongside the aorta, passes through the diaphragm, enters the right atrium of the heart. So the vena cava is not a single vessel formed in one step, instead it is the result of convergence. Okay, so the internal iliac vein, external iliac vein together will form the inferior vena cava. Okay, subclavian vein, internal jugular, brachiocephalic will form the superior vena cava. This is clear. External from the limbs, internal from the pelvic organs and gluteal region. Fifth lumbar vein. So the right and left common iliac veins merge anterior to the fifth lumbar vertebra to form inferior vena cava. Whereas the SVC, the right and left brachiocephalic veins join posterior to the inferior border of first right costal cartilage. So first costal cartilage to form superior. This one is fifth lumbar vertebra. Okay. Now, Does it
Development is a really broad topic. To give you a help,
inferior vena cava?
the term inferior vena cava seems to be a misspelling or a less common way of referring to the inferior
Never
vena cava.
listens properly.
Therefore, I will explain the embryological development of the inferior vena cava. The development of the inferior vena cava is a complex process that occurs between the 6th and 8th weeks of intrauterine life. It involves the formation, regression, and fusion of three pairs of embryonic veins. 1. Posterior cardinal veins. These veins initially drain blood from the caudal part of the embryo. 2. Subcardinal veins. These veins appear later and drain the developing kidneys, gonads, and adrenal glands. 3. Supracardinal veins. These are the last set of veins to develop and drain the body wall and limbs. The inferior vena cava forms through a series of anastomosis, connections, and regressions among these three venous systems. The right-sided veins generally persist and contribute to the formation of the IVC, while their left-sided counterparts mostly regress. Here's a simplified overview of the process. Initially, the posterior cardinal veins are the dominant drainage pathway. The subcardinal veins develop and form connections with the posterior cardinal veins. The portion of the IVC that drains the kidney, suprarenal segment, is derived from the right subcardinal vein. The supracardinal veins appear and also anastomose with the other cardinal systems. The portion of the IVC below the kidney, infrarenal segment, develops from the right supracardinal vein. The hepatic segment of the IVC, which drains blood from the liver, develops from the right vitelline vein, another embryonic vein system. Through a series of fusions and regressions, the mature inferior vena cava is formed on the right side of the abdominal aorta, draining blood from the lower limbs, pelvis, and abdomen back to the right atrium of the heart. Variations in this complex developmental process can lead to congenital anomalies of the inferior vena cava, such as a left-sided IVC or a double IVC.
So this is the process. So we have three pairs. Okay, posterior cardinal veins. My pen is not working. Posterior cardinal veins. So these veins initially drain blood from the caudal part and tail part of the embryo. Then subcardinal veins. These veins appear later and drain the developing kidneys. Okay, kidneys and all. Supracardinal is veins to develop and drain body wall and limb. Okay, body wall and limb. Supracardinal. Okay. So initially the posterior cardinal veins are the dominant drainage pathway. Cardinal veins were the posterior cardinal veins were the dominant one. Then the subcardinal veins develop and form connections with the posterior cardinal veins. A portion of IVC that drains the kidneys, suprarenal segment is there from the right subcardinal vein. The supracardinal veins appear and also anastomose with other cardinals. The portion of IVC below the kidneys develops from right supracardinal veins. That drains the kidneys, suprarenal veins derived from right subcardinal Supracardinal veins appear and also with the other cardinal portion below the kidneys. Below the kidneys is infrarenal. There are some right supracardinal, okay, right supracardinal, right subcardinal. The hepatic segment of IVC drains blood from liver develops from the right vitelline veins and the embryonic vein system. The series of fusion regression mature and female combines from the red side. This is not given in detail. We'll see from some other book. So developmental anomalies of SVC superior vena cava is left superior vena cava is formed in left anterior cardinal and common cardinal veins persist and the right ones obliterate. Left superior vena cava opens its right atrium to the coronary side. Double superior vena cava occurs due to persistence of left anterior cardinal veins. But here the answer is due to persistence of both supracardinal and subcardinal veins. Double IVC is formed. That's okay. So, anomalies of SVC, let's see, because sometimes from these topics we will ask the question. It's better to go through this. SVC is formed when left anterior cardinal and common cardinal veins persist and right ones obliterate. Left SVC opens into right atrium to the coronary sinus. Double superior vena cava occurs due to persistence of left anterior cardinal veins. The right SVC opens directly into right atrium while left to coronary sinus. IVC, absence of inferior vena cava above renal veins occur when the anastomotic channels between right subclavian vein and right hepatocardinal channels fail to develop. Double inferior vena cava is formed below renal veins. It consists of both subcardinal and supracardinal veins below the kidney. Left inferior vena cava is that is infrarenal part of IVC is from left side instead of right. Pre-diabetic IVC is formed when infrarenal part of IVC is also subcardinal instead of cardinal, supracardinal veins. Okay, jelly formed around the heart tube during early development contributes to the formation of jelly formed around the heart tube during early development contributes to the formation of myocardium. So jelly formed around the heart tube during early development contributes to the formation of the myocardium, which is the middle layer, which is very thick, and there's a muscular layer which is responsible for pumping. Okay. So dorsal mesocardium forms transverse pericardial sinus. Somatopleuric mesoderm forms the parietal pericardium. Splagnopleuric mesoderm from myocardium and conductive system. Okay. Neural crest cells from subpulmonary inferior cardiac jelly from the cardiac tissue membrane. The retina is an outgrowth of the. So the answer is B. Retina is the outgrowth of the diencephalon. Retina is the outgrowth of the diencephalon, not meson, not telen, not pons. Diencephalon. So diencephalon forms optic cup and stalk between the thalamus, hypothalamus, epithelium, fibula, and third ventricle. Retina develops from the walls of optic cup. The outer thinner layer becomes retinal pigmented epithelium, inner thicker layer forms the layer of retina. So inner one is an inner thicker layer forms the neural layer and outer thinner layer will form the pigmented epithelium. Foramen is called prosencephalon. Okay, consists of telencephalon, anterior or rostral part, and diencephalon, posterior part or caudal part. Arch of aorta develops from which aortic artery? Fourth left. Okay. Arch of aorta develops from which aortic artery? We see this in detail.
While the initial query is a bit ambiguous, it
It will read, I'll mark, okay.
The arch of the aorta undergoes a complex developmental process during embryogenesis, arising from several structures derived from the pharyngeal arches and the aortic sac. Here's a breakdown. 1. Aortic arches. During the fourth week of development, 6 pairs of pharyngeal arches develop. Each arch contains an artery, a cranial nerve, a cartilage rod, and a muscular component. These aortic arches connect the aortic sac, the most cranial part of the truncus arteriosus, with the paired dorsal aorti. However, not all 6 arches persist or contribute directly to the adult aortic arch. 2. Contribution to the aortic arch. Aortic sac. The portion of the aortic arch proximal to the brachiocephalic trunk originates directly from the aortic sac. Left fourth aortic arch. The medial part of the aortic arch, located between the brachiocephalic trunk and the left common carotid artery, develops from the left fourth aortic arch, dorsal aorta. The portion of the aortic arch distal to the left common carotid artery is derived from the left dorsal aorta. 3. Fate of other aortic arches. First aortic arch. Mostly regresses, but a remnant contributes to the maxillary artery. Second aortic arch. Primarily regresses. Remnants form the stapedial and hyoid arteries. Third aortic arch. Forms the common carotid artery and the proximal part of the internal carotid artery. Right fourth aortic arch. Develops into the proximal part of the right subclavian artery. Fifth aortic arch. Either never fully forms or regresses completely. Sixth aortic arch. The right sixth aortic arch becomes the proximal part of the right pulmonary artery. The left sixth aortic arch forms the proximal part of the left pulmonary artery and the ductus arteriosus, which becomes the ligamentum arteriosum after birth. In summary, the arch of the aorta is not derived from a single aortic arch, but is a composite structure originating from the aortic sac, the left fourth aortic arch, and the left dorsal aorta. The other aortic arches contribute to different arteries in the head, neck, and pulmonary circulation.
Seems so, if you see this, first one will regress, but it will form the maxillary artery. Second one regress, remnants stapedial and hyoid artery. Third one, common carotid artery, okay, and proximal part of internal carotid artery, ICA and CA, CCA, common carotid artery. Fourth aortic arch develops into proximal part of right subclavian artery. So fourth aortic arch, right one. So fifth aortic arch, so this is not fully formed, not regressive. Okay. Okay, sixth one, proximal part of proximal part of left pulmonary artery and ductus arteriosus. So if you see here, and so left of fourth one is the answer. Muscles of the third arch. So we'll ask this, tell us some trick. So muscles of a third arch is the stylopharyngeus. Stylopharyngeus is the muscle of third pharyngeal arch. Let's see what it says.
The pharyngeal arches, also known as branchial arches, are structures that form during the embryonic development of vertebrates, including humans. These arches are a series of paired bulges on the lateral surface of the developing head and neck region. They are crucial for the formation of various structures in the head and neck, including bones, muscles, nerves, and blood vessels. In humans, there are typically 5 pharyngeal arches that are well defined, numbered 1, 2, 3, 4, and 6. The fifth arch is rudimentary and not always considered. Each arch contains all 3 germ layers, ectoderm, mesoderm, and endoderm, and is associated with specific cranial nerves and aortic arch arteries. Here's a breakdown of what each arch contributes to. First, pharyngeal arch, mandibular arch, skeleton, mandible, maxilla, zygomatic bone, malleus and incus, middle ear ossicles, sphenomandibular ligament, muscles, muscles of mastication, masseter, temporalis, medial and lateral PT ergols, anterior belly of digastric, mylohyoid, tensor tympani, tensor vermicalis, nerve, trigeminal nerve, CNP, artery, maxillary artery, second pharyngeal arch, hyoid arch, skeletal, state, middle ear ossicle, styloid process of the temporal bone, stylohyoid ligament, lesser horn and upper part of the body of the hyoid bone, muscles, muscles of facial expression, stapedius, stylohyoid, posterior belly of digastric, platysma, nerve, facial nerve, CNBII, artery, stapedial artery, usually regresses. Third pharyngeal arch, skeletal, greater horn and lower.
Sorry, this
The pharyngeal arches are a series of paired structures that develop in the neck region of the developing embryo. These arches are crucial for the formation of various structures in the head and neck, including bones, muscles, nerves, and blood vessels. In humans, there are typically 5 pharyngeal arches numbered 1, 2, 3, 4, and 6 arch 5 regresses during development. Each arch contains a cartilaginous rod, a muscular component, a nerve, and an artery. These components differentiate to form specific adult structures. Here's a common mnemonic to help you remember the nerves associated with each of the pharyngeal arches. 1, 2, 3, 4, and 6 get trigeminal, facial, glossopharyngeal, vagus, vagus again. First pharyngeal arch, trigeminal nerve, CNV. Second pharyngeal arch, facial nerve, CN7. Third pharyngeal arch, glossopharyngeal nerve, CNIX. Fourth pharyngeal arch, vagus nerve, CNX. Superior laryngeal branch, sixth pharyngeal arch, vagus nerve, CNX. Recurrent laryngeal branch. This mnemonic focuses on the primary nerve supply for each arch, which is a key.
बहुत अच्छा नहीं बोले कि
Would you
बहुत
like to know about the derivatives of
बकवास
each of these arches as well?
सारी trigeminal, then facial. First pharyngeal is trigeminal, then second is face, third is glossopharyngeal, fourth is vagus nerve, superior laryngeal branch, sixth is recurrent laryngeal branch. Can be the next question. Recurrent laryngeal branch.
That's a great question. Let's delve into a more detailed description of the pharyngeal arches and their derivatives. During the 4th week of embryonic development, neural crest cells migrate into the head and neck region, leading to the formation of 6 pharyngeal arches. However, the 5th arch regresses and doesn't contribute significantly to adult structure. Each arch is covered externally by ectoderm and internally by endoderm, with a core of mesoderm, specifically paraxial mesoderm and lateral plate mesoderm,
We'll discuss
and
tomorrow.
neural crest cells. Each pharyngeal arch